Thank you very much, Carlos, for agreeing to give a Build a Cell seminar, and you can get started.
All right, Kate, well, thank you very much for the invitation, and I'm glad to be here today. My seminar is going to be a little bit different compared to the recent seminars that I've attended as part of the series. My background is in physical chemistry, really, and most of the work that we've done in my group has been on understanding interfaces. That includes lipid membranes, of course, as well as surfactants. But recently, since COVID, we started collaborating with a group at UCSD to understand environments in synthetic membranes. So that is the work that I'm going to be presenting today during this seminar. And as I said, since this is a physical chemistry heavy talk, I have a lot of background slides on the techniques that we use, and I'm happy to answer questions at the end of the talk. Before I start, let me just acknowledge the people who did this work. This is my group currently at the University of Texas at Austin, and my student Kong Zhu is the one who's been spearheading this project on synthetic organelles. This was funded by Muri from the Army Research Office, ARO. That is, we're part of a large collaborative team on building synthetic cells. And our part of this is the synthetic membranes or synthetic organelles. I should mention Kong is a physics student in my group who's getting ready to graduate within the next semester or so. Just to give you an overview of the types of projects that we have going on in the group, I don't want to kind of dwell into any specific project for too long. Since we are a biophysics group, but really understanding the physical chemistry of biological systems, we have quite a few things that we're working on. We've done, as I said, we've done a lot of work on membrane interfaces. We're also working on transmembrane proteins, understanding the mechanisms by which proteins fold into lipid membranes. We have done some recent work on ion binding. Specifically, we have a focus on lanthanide ions for the purpose, not necessarily biological, but for the purpose of materials recovery and so on. As you know, there's been a lot of push on critical minerals recently, and so we got into this research area. And other projects are on understanding confined environments, such, for example, the environments you would see in the cytosol of a cell. As you can imagine, the dynamics of the water molecules, the way that water molecules interact with each other inside of a cell is quite different compared to bulk water. And the reason for that is because water has extended hydrogen bond networks. I'm going to talk a little bit about that later in the talk. But due to this confinement, the hydrogen bond networks are disrupted, which then creates environments that slow down the hydrogen bond dynamics or the switching process of water. And so because of that, it is important to understand how the solvent is in a cell. So for example, here is something like a red blood cell in comparison to, let's say, a buffer solution. And then we've also done a fair amount of work on surfactants and reverse micelles and so on. So because of that, because of this background that we've had, we were invited to join this team on the synthetic organelles, which in my view really combines some of the knowledge that we gained from studying natural lipids and surfactant interfaces. So let's start with what are organelles and what are the challenges that we have in order to design the synthetic organelles. As we all know, compartmentalization is an important principle in biology, and organelles are one of the main mechanisms by which cells compartmentalize and organize biochemical processes, such as chemical reactions and so on. But not only that, an organelle is not just a barrier to for these processes, but it provides these dense membrane networks with very large surface areas, right? If you think, for example, the ER or mitochondria, they have all sorts of different structures within this organelle, sorts of different folds in the membrane. And this is important because they have the ability to sequester membrane proteins as well as soluble proteins. and then control the diffusion of small molecules that participate in these biochemical processes. And therefore, designing artificial organelles, like I wrote here, requires being able to control the membrane morphology and the interfacial properties. So if we want to build an organelle, build a cell, we need to come up with lipid compositions that have very well-defined interfacial properties. Now, you might be wondering, why do we care so much about interfacial properties? Well, we care because biological processes happen at the interface. We've all learned that about 1/3 of the genome codes for membrane proteins, right? So these membrane proteins live in this interfacial environment between the hydrophobic region in the core of the membrane and the hydrophilic cytosol, right? And so we need to be able to first understand and then control this interfacial environment. So now, if we want to build an organelle, we have to get away from vesicles. We know from our studies that vesicles do not capture the complexity that we require for certain biochemical processes. And so the idea here is we would start, we start with something simple, something where we understand, something that's being used a lot in biophysical studies, such as, for example, a lipid vesicle, and how can we start increasing complexity to build something that maybe resembles a biological lipid membrane, right? And not only how we build it, but how do we study? How do we know that we have the right morphology? How do we know that we have the right interfacial properties? How do we know that proteins that are embedded in these environments have environments or live in a similar environment as they would in a natural lipid membrane? And then on top of that, what is the minimal set of lipids that we need to recreate this complexity? If you think about lipid membranes in cells, we have hundreds of different species, hundreds of different lipids. Do we need to have that complexity? Do we need hundreds of lipids, or can we recreate similar environments with maybe a much smaller subset of lipids, right? And in terms of studying this, there are many challenges, some of them which I've highlighted here. Since these are such heterogeneous environment, studying this requires species-specific probes, right? If we have hundreds of lipids, can we study the environment around one individual species so that then we can recreate similar environments artificially? And in addition to that, we need to have probes, molecular probes, that are sensitive to the local environment. And so this is one of the key challenges in the techniques that I'm going to be describing to you in a little bit, is finding the right probes to be able to access the environment. And then in addition to that, in terms of the both experiments and in terms of modeling, we have to deal with multiple timescales. If you look at the timescales of dynamics in lipid membranes, they span 12 orders of magnitude, from picoseconds hydrogen bond dynamics, which I'm going to be talking about today, all the way to things like vesicle formation and processes that happen on much, much longer timescales. So we need to have experimental probes that give us access to this wide range of timescales. Now, in my lab, we do infrared spectroscopy. IR spectroscopy is an ideal tool to be able to probe the environment around molecules. So here I'm going to describe to you how IR spectroscopy works and why it is such a powerful tool. You've probably heard about IR spectroscopy, mainly probably from like a synthetic organic type chemistry context, where you say, well, I'm going to measure the IR spectrum of a molecule to see what functional groups are present in that molecule. And so that's not how we use IR spectroscopy in my group. We use spectroscopy to be able to tell us about the environment that this molecule is in. And so just to give you an example, this is a very small molecule, ethyl acetate. And this molecule has a carbonyl. So if you look at this carbonyl vibrational mode in the experiment, what you see is exactly 3 different peaks. And you might be wondering, well, I have one carbonyl. How come? And when you measure the sample in the solution, you see 3 different frequencies, right? I only have one carbonyl. I only have one type of molecule. Well, the reason why you see 3 different frequencies is because this carbonyl can hydrogen bond with, in this case, water or whatever hydrogen bonding solvent you put this molecule in. And so because of that, what that tells you is that by measuring the areas of the different peaks, you can actually get at the hydrogen bonding populations. So just to show you an example of that, this is exactly this molecule ethyl acetate in methanol. And you can see the spectrum that you see in the dots is the measured spectrum, and you can fit it almost perfectly to a combination of 3 Lorentzian functions. And then the populations of the different hydrogen bonds. So this is 0 hydrogen bonds, meaning the carbonyl that's not hydrogen bonded to anything. 1 hydrogen bond would be 1 OH interacting with this carbonyl. 2 hydrogen bonds would be 2 OHs. In the experimental example is methanol, but here in the cartoon is water. What's powerful about this is that you can take the areas of those different peaks and you can get the populations of the different ensembles, right? So it tells you immediately about the environment around this molecule. And then you can modify, for example, the solvent properties. Instead of methanol, you can choose different alcohols, and those ratios will vary depending on the environment. In the case of lipid membranes, natural lipids, there's many different vibrational probes that you can use in a similar way. So this is a cartoon here showing some of the vibrational modes that we've used, and as well as other groups have used in the context of IR spectroscopy to be able to probe different properties or different environments within the lipid membranes. In particular, as you know, phospholipids have these ester carbonyl linkages, and these are very useful because these carbonyls sit right at the interface between the hydrophobic center of the membrane core and the hydrophilic environment around it, right? So that's we've done a lot of the previous work on natural membranes on this using these carbonyl probes to look at the interfacial environments. In the synthetic lipids, we have different vibration. In addition to that, you can actually probe the water dynamics directly. There's different spectroscopic tools that enable you to look at the OH vibrational modes in water. We don't do that in my lab because it requires a specialized setup. And then you can also look at the phosphate groups in phospholipids, or if you have, for example, proteins that are embedded in the membrane, you can probe the backbone carbonyl groups. Those have been very widely used in the spectroscopy community to probe the secondary structure of these proteins in the membrane. Now, one thing that I want to mention briefly today, it's not the main focus of this talk, but I do want to mention it, is that there's a new technique called surface-enhanced infrared absorption spectroscopy. And this requires, if you're familiar with IR spectroscopy, this requires putting some gold nanoparticles on an ATR crystal. An ATR crystal is a total internal reflection crystal. And what that does is it amplifies the electric field from the infrared light at the interface between the crystal and the solvent, so you can get very, very high sensitivity. So we just started using this technique in my group. What that enables us to do is we can take a single lipid bilayer, deposit it on the ATR crystal, and then measure spectra directly of proteins inside of this bilayer. So it significantly enhances the sensitivity of infrared absorption spectroscopy such that we can get this monolayer in quotes. We can measure single lipid bilayers. And just to demonstrate that, here is some spectra that we measured of a little helical peptide that folds and inserts into the membrane as a function of pH. And you can get a very nice titration curve with this technique. But not only that, it's easy to see when a peptide is folded or not folded, right? That you can use CD spectroscopy or other techniques. But what we get is the full infrared absorption spectrum of this peptide. So we can get access to not only the structure, but also the environments around this peptide. So we're excited to start using this technique in the future. Here's the picture. It's essentially just an FTIR. This is the instrument, and unfortunately, you cannot see this Cyrus crystal, but it's inside of the instrument. Now, everything I mentioned so far is about the environment, the structure. What is these lipid membranes? What environments are they experiencing? But what we're interested in is also the dynamics. How are these environments evolving in time? Okay. And so what that means, here's a little movie that I made from a molecular dynamics simulations. What that means is we need to be able to probe the changes in the environment as a function of time. Now, when I say time, that means that there's got to be some characteristic timescale that we need to be able to access with our experiment. So in the case of molecules, that characteristic timescale is picosecond timescale. Picosecond is 10 to the minus 12 seconds. And that is typically the lifetime of hydrogen bonds. I'm showing here a movie from a molecular dynamics simulation showing this phosphatidylcholine lipid and showing these ester carbonyls that sit right at the interface between the hydrophobic and the hydrophilic environments. And you can see that there's water that's penetrating all the way down to the carbonyl region. So we've done a lot of spectroscopy on this carbonyls because the amount of water penetration into this region depends on the head group, depends on the tails, depends on the packing efficiency of these lipids, depend on what else you have. For example, if it's a highly crowded membrane, the dynamics are going to be different compared to a simple vesicle made up of just lipids with no protein. So that gives us, as I said, this is one way to probe the changes or the time evolution of the environment within these interfaces. So for that, we use two-dimensional infrared absorption spectroscopy. If you're familiar with 2D NMR, this is the infrared analog of 2D NMR. If you're not familiar with 2D NMR, that's okay. I'm going to explain this in just 1 second. And what you need to understand here for this technique is that now we have two frequency axes. That's what the two-dimensional means here. It's not spatially resolved or anything, but it's spectrally resolved. So we have two different frequency axes. We have an excitation frequency along the horizontal axis, and then a detection frequency that we plot along the vertical axis. In addition to that, we can vary the time delay between the excitation pulses and the detection pulses in the spectroscopy, so we can actually see how the different systems evolve in frequency space. And so the way to think about it is, now imagine that you have a broad peak in your spectrum, or it could be multiple peaks, like I showed you in the previous slides where we saw 3 different peaks. So what that means is that you have a distribution of environments that the molecule is experiencing. And what we want to know is, how quickly do these environments randomize? How quickly do these environments exchange in our frequency space? So what that means is, if you don't allow any time for the system to evolve, in other words, we come in with a short laser pulse, excite the molecule, and for example, a molecule that's on this side of the peak means it absorbs towards higher frequency. When you excite that molecule, then the next pulse stimulates emission from that molecule, and it's going to emit at the same frequency because the system hasn't had time to evolve, right? You can think of our carbonyl example. If it's hydrogen bonded, you excite the carbonyl, it has a given frequency, and when you probe it, it has the same frequency because it remains hydrogen bonded. However, if you allow time for the system to evolve, maybe you excite the carbonyl when it has a hydrogen bond, that hydrogen bond breaks, and when you probe it again, it's going to have a different frequency. And so what that means is we're losing correlation between the excitation and detection frequency as we increase that time, which is called the waiting time between the excitation and detection pulses. What we measure in this technique is a frequency fluctuation correlation function. So what that means is, if you have an environment where the fluctuations in the frequency are fast, that correlation functions decays fast. And if you have an environment such as, for example, in a lipid membrane, that correlation decays a lot slower because now the hydrogen bonds are more long-lived. And so that's the powerful thing about this technique, and it's what enables us to measure the dynamics, like I said, in either interfacial environments or confined environments, and understand how the environment is linked to how molecules evolve within these environments. Here is a picture. I always like to show pictures of the lab. This is a picture of the laser setup that we have here at the University of Texas at Austin. This is an outstanding student in my group, Rina Al-Mualem, and she's here changing the sample. This is where the sample goes. but it's a whole laser table that is used for generating the infrared pulses and then bringing them all together at the sample and then detecting them after the sample. So that's the whole system. We built this in 2017. And now we have two setups in my group, two independent 2D IR setups. Now let's get into synthetic organelles. Let me just check the time here. Okay, we're good. Let's get into synthetic organelles. As I mentioned at the beginning, this is a collaboration with the Devaraj group at UC San Diego. And the idea here is, can we use purely synthetic lipids that don't exist in nature to be able to control the morphology of the organelles, right? So we like to use this analogy of having different Lego pieces and putting them all together to be able to create something that maybe resembles an organelle. Of course, this is a very ambitious goal, and I'll tell you up front, we're not there yet. But we think we've made some steps in understanding how the structure of the lipid, as well as how the composition is able to determine the morphology of the structures that we make. So this is just maybe a little basic slide, but I wanted to remind everyone that when it comes to lipids, there are really two main things that you can control. One is you can control the acyl chain morphology. So this is by controlling the unsaturation, where you put double bonds and things like this. And you know that if you have fully saturated acyl chains, they're able to pack a lot better. So that's kind of like butter. And so what that means is the fluidity is a lot lower compared to if you have unsaturated lipid tails, which are not able to pack as tightly as saturated, and you get something like olive oil, for example, right? And then the other thing that we can control is the head groups. So in this case, I have two different lipids. These are not synthetic lipids. This is DPPC and DPPE. And one of the things that we found with studying these natural lipids is even smaller changes can have quite significant effects in terms of membrane properties. So here is DPPC, which has these 3 methyl groups in the choline chain here, right? And DPPE, instead of the methyl groups, you have 3 hydrogens. These two lipids, even though they look more or less similar, right? There doesn't look like you're making a big change here, have quite different interfacial properties. And the reason for that is DPPE now can hydrogen bond. So you can see this is NH3+. So this can actually hydrogen bond with other lipids. You can hydrogen bond with the phosphate groups. You can even hydrogen bond to the carbonyl because the lipid head group folds over and interact with other head groups. So the properties of the membrane are quite different. I'm not going to talk about this project. We published this a couple of years ago now. But I did want to emphasize that these are the kind of the two knobs that we can control, but it's very difficult to link the structural changes to changes within the environment. Now, in the context of artificial lipids, we have decided to study this glycolipids that the Devaraj group has been making. These are purely synthetic lipids. And one of the interesting things is that depending on the unsaturation of the tail, you can get quite different phases. So here I have 4 different lipids that were made by the Devaraj group. The one that I'm going to describe today in the next few slides is this GOA. So it has this galactopyranoside as the head group, and it has 1 unsaturation. This oleoyl is the fatty acid that is used to make this lipid. And the linkage here, instead of being the ester carbonyl that is found in natural phospholipids, it's an amide. So you can see this amide group here, CONH. But in terms of the spectroscopy, it's very similar to what we have observed in the natural lipids. And in fact, we can take all of the techniques that we've developed for the natural membranes and apply them to the synthetic membranes. Now you can see, right, this illustrates a point that I was trying to make, that even small changes can have significant effects on the morphology. So for example, if you compare GOA, I'm going to talk about this phase diagram. but it forms, it can form vesicles or it can form a sponge phase compared to GPA. So GPA just moves one of the hydrogen bonds to a slightly different position. And this lipid actually just aggregates. It doesn't form vesicles or anything. It just forms aggregates in solution. GLCOA, this is changing the position of one of the chirality of one of the OH groups here, right? That forms these nanotubes, nanotube structures. Otherwise, it's exactly the same as GOA. And then GLA is adding another double bond here. So this is linoleic acid. This here can actually form this large vesicle structures that were characterized by the Varage group. So even just changing, it's not as simple as I said, oil versus butter, but even changing the position of the double bond can have quite significant effects on the morphology. So for our study, we decided to use a combination of GOA, this lipid here that I introduced in the previous slide, with IGPAL. IGPAL is also known as Triton X100. So this is also an artificial surfactant that has a long PEG chain in the head group. And the question is, can we make structures that have similar organization as, for example, some of the cell organelles that I described earlier, right? And so these two lipids, I'm going to show you the phase diagram in the next few slides, but if you mix them together, you can actually make this sponge phase vesicles. These are relatively large. They're about 10 to 15 microns in size. The scale bar here is 15 microns. So you can easily see them in the microscope. And the morphology inside of these structures, these droplet structures, is analogous to an organelle. They form these channels. This is actually called a sponge phase because it resembles the morphology that you would see in a sponge, where you have these very small channels and very large surface areas. The size of these channels here has been characterized with X-ray diffraction, is on the order of about 3 to 5 nanometers. So comparable to what you expect for something like mitochondria or ER or some of the other organelles in the cell. And the Varage group in this paper that they published in 2020 did a lot of different studies on this synthetic organelles, or the sponge droplets, as they call them. And you can put all sorts of different dyes, staining dyes. They all go in there. You can even put proteins here. These are different proteins that interact with the carbohydrate head group. So, for example, galectin here is, as you might know, is a type of protein that binds the head group, right? In this kinds of lipids. You can also express proteins, and this leg A, for example, goes straight inside of this droplets, or you can do some more kind of standard chemistry. So for example, here, nickel NTA labeled lipids, you can add those to the droplets, and then you can put a his-tag protein and it goes inside of the droplet. Sorry. And you can also change the charge balance. So CTAB is a positively charged surfactant. It's not a lipid because it's not found in nature. So it's a surfactant. You can put this surfactant in the vesicle, and you can put an oligonucleotide, which is negatively charged. And you can see here in these microscopy images that it actually is localized to these droplets. Or you can do biotinylated lipids and add streptavidin, and you can see the same effect. So they've been able to show that by making this artificial organelles, you can actually do quite a lot in terms of controlling how you can localize proteins to these environments. Let's see, next slide. There we go. Now the question is, what do we know about the composition, or how the composition is reflected on the morphology, right? And so in the Varage group, they characterize this phase diagram, which here it shows this is the concentration of GOA, which is our artificial lipid, and the concentration of IGPAL, which is the surfactant, the co-surfactant in these membranes. And depending on the ratio, you can either get vesicles, you can get this droplet phase or sponge phase, which we're interested in, or you can get micelles. And the The sponge phase is important because, like I said, these are X-ray diffraction measurements. This is inverse space here, inverse angstroms. It has this distribution of different channels that you don't get in either the vesicle phase or the micelle phase. You can see in the micelle phase, you get this very sharp peak, which means it's relatively uniform. Whereas here, this peak is broad. That means that there's a large distribution of sizes within these channels. Now, where we come in with our work is we want to understand how the composition determines those interfacial environments, not just the morphology that's been characterized already, but we want to understand how does the composition modify those interfaces. And so to do that, we use 2D infrared spectroscopy, as I described previously. We can take the 2D spectra and then we can take a cut along the diagonal. I'm not going to talk too much about the 2D spectra themselves. We understand those very well. You can think of this diagonal slices like an FTIR or an IR absorption spectrum qualitatively. And then, as I mentioned in the previous slides, what we can do is we can fit the spectra to a combination of different Gaussians. And from those Gaussians, we can actually understand we can quantify the different hydrogen bond populations within these different compositions. So that's shown here in this diagram. This is the average number of hydrogen bonds as a function of the Eigepal to GOA ratio. So as we go from the vesicle phase to the sponge phase to the micelle phase. And the blue curve here shows the experimentally measured hydrogen bond populations. As you can see, in general, there's a decrease in the number of hydrogen bonds as you increase the amount of GOA. So to us, this was interesting and maybe a little bit surprising at the beginning. We didn't really understand why it would be that as you add more lipids, remember, we're probing these carbonyls here, right? So as you increase the amount of lipids, the carbonyl becomes more dehydrated. There's fewer hydrogen bonds. And so to understand that, we turn to molecular dynamics simulations. One of the strengths of our group is we're primarily an experimental group, but we do a fair amount of simulations that enable us to interpret the experiments. In the MD simulation, these are all atom MD simulations. You pack all the molecules into the box. You set up some sort of description of the forces. In this case, we use the charm force field, which is one of the most used force fields in this community. And you let the molecules organize themselves as they would, right? And then we can understand how the environments change as a function of the composition. So we run several simulations with different ratios of the GOA and our surfactant. So this is what the surfactant looks like. Remember, we have this kind of long peg chain here as part of the head group. When you look at the structure here in the surfactant, it's not always obvious when you put it, but here you can see 9 to 10 repeats, right? So when you show the full 3D structure of the surfactant, this is what it looks like. And this is extending out into solution. And here's GOA. The carbonyls here, this is a carbonyl here shown in red, and the rest of the OHs are also shown in red. This is a head group, and that's how it packs into the box. And then we want to see how many hydrogen bonds are being made to this amide carbonyl that we have here. And so for that, we use what's called a geometric descriptor of the hydrogen bond. That means that if you have two oxygens within a certain distance, in this case, 3.5 angstroms, and if the cutoff angle is below a certain, if the angle is below a certain cutoff, then you count that as a hydrogen bond. Otherwise, you don't count it as hydrogen bond. And so what that enables us to do now is we can actually calculate the number of hydrogen bonds from these molecular dynamics simulations. So one thing we can do with the MD simulation, which we cannot do with the experiment, is we can actually separate the hydrogen bonds to water versus the hydrogen bonds to other head group, right? So coming back here to the structure, you can see that there's a lot of OHs that are part of the head group here. And because of that, this carbonyl can hydrogen bond to the neighboring lipid OH In the experiment, we did not see those as separate from water because we only see a frequency shift from a hydrogen bond. Whether that OH is from a head group of a neighboring lipid or from water, we don't know. However, in the simulation, we can actually separate them out because we have the structures, we have the trajectories, we know how all these molecules interact with each other. So we can apply the geometric descriptor to an OH from the head group versus from an OH from water. And what we see here is that, first of all, that the simulation, which is shown here in blue is the experiment, sorry. Red is the water to GOA hydrogen bonds, and then this black is the GOA to GOA hydrogen bonds. And so what we see here is that there's a decrease in the number of hydrogen bonds in the simulation as well, which was good because that means that qualitatively we are in good agreement with experiment. Okay, so now the question is why? Why do we see this dehydration as we add more of this GOA? And we can do additional analysis from the simulation. So for example, we can look at this density maps. This shows the density of the different components as a function of the composition. And so on the horizontal axis, you have the distance from the center of this bilayer. So this is the center of the bilayer. And then you can see what is the density of the different components. So as you increase, essentially, as you increase the amount of GOA, so here as you increase the amount of IGPAL, what you can see is that the lipid membrane becomes a little bit more expanded, right? So the tails are packing better together when you increase the amount of this co-surfactant in the lipid membrane. And so we were excited to see these morphology changes that are happening as a result of the different compositions of the membrane. And that already gave us a clue as to what's happening here. What's happening is essentially the lipids are packing closer together as you increase the amount of co-surfactant. And this additional packing prevents water from coming in and hydrogen bonding with the carbonyls. Remember, the carbonyls are sitting pretty far in, right? Oh, sorry, I went one slide too far. I keep coming back to this slide. But the carbonyls are pretty far in. After the carbonyl, everything else is hydrophobic. So as you pack these lipids together in more closely, then less water is going to be able to penetrate into this interfacial region where the carbonyls sit. We've done a lot of other analysis as well, where we actually look at the packing of the PEG chain as well as the acyl chain ordering, and all the analysis essentially tell the same story, which is as you increase the amount of IGPAL, these lipids are packing better together. This is, let me just describe these figures here. This is the angle of the carbon-oxygen. If you think of it as the carbon-oxygen bond in this PEG tails here, right, in comparison to the normal of the bilayer here, right? So if that angle is 0, that means that these PEG chains are packing very closely together. If there's a large distribution here, as you can see in the blue, where you have a very little IGPAL, that means that there's a lot of disorder in these PEG tails. And you can actually, sorry, let me go back. And you can actually even see it here that what happens is as you increase the amount of our co-surfactant, of this IGPAL co-surfactant, these PEG tails are actually seem to stack with each other. They're starting to pack together very closely. So this suggests that there's this microscopic phase separation that is happening in the membrane. In other words, you add IGPAL and it just doesn't get uniformly incorporated into the membrane, but it forms patches that are enriched in IGPAL and patches that are enriched in GOA. And so now if we start thinking about the dynamics, this is the same slide that I showed before. We use 2D spectroscopy to measure those hydrogen bond lifetimes as I described in the beginning. Here is the measurement, which is shown in red. This is the 2D IR lifetime of the hydrogen bonds, and the simulations are shown in blue. And you can see that qualitatively, the two axes are slightly different. That means that the simulation is showing a little bit faster hydrogen bond dynamics compared to the experiment, but qualitatively, they're both in very good agreement. Now, for the sake of time, I'm going to skip the last couple of slides. I wanted to describe the computational models, but that's not super important for this talk. Instead, I'm just going to go back to the kind of the last analysis that we did, which is what is the structure of water around these interfaces as a function of the composition. There are many ways to describe water, but one of them is using this tetrahedral order parameter. This is widely used in the community, and it is a way to measure how ice-like, in quote, the structure of liquid water is. Okay, so as you know, water can donate 2 hydrogen bonds and it can accept 2 hydrogen bonds. So it assumes its natural tetrahedral structure. In ice, this is an actual perfect tetrahedron. So 109.5 degrees are the angles. In liquid water, what you can do is once you run the simulation, you can take a water molecule, find its 4 nearest neighbors, and then project that structure onto a perfect tetrahedron. This is the mathematical definition of that. So you basically measure the cosines of all the angles and you project them onto a tetrahedron. So if you have ice, that projection would be 1. So the maximum value of this parameter is 1. And if you have a completely random structure, on average, that projection would be 0. Okay. And so this here shows the distribution of these parameters for liquid water. So lower means an ideal gas, higher means ice-like tetrahedral structures. And this is well known in the community. Basically, you see 2 different distributions. You see what's called high density water. This is here, and low density water, or more ice-like water. And now what we want to know is how does this parameter change for the interfacial water molecules in this synthetic membranes, okay? And so we can compute that. Here is this average order parameter computed from the simulation as a function of the amount of co-surfactant that we add. And here, so each of the different curves is each of the different compositions that we have. And the horizontal axis is the distance from the center of the bilayer. Obviously, we cannot start at 0 because there's no water in the center of the bilayer, so there's nothing to measure. But as soon as you get about 0.5 nanometers away from the center of the interface, you have enough water that you can get these tetrahedral structures. And what you see is that interfacial water is more disrupted compared to bulk water. Okay, that's something that we already knew. It's nothing new. But we had done this similar studies in natural lipid membranes. But what we see here, this is 1.0 is the highest concentration of co-surfactant, 0 is just the pure GOA. That when you add this co-surfactant, it actually disrupts the structure of water. That's kind of what you see as the curves get lower and lower, right? So that means less and less structure. This order parameter, if you get close to 1, is ice-like. If you get close to 0, it's kind of gas-like. So it's highly disordered at the interface, and it becomes more bulk-like away from the interface. But this co-surfactant actually is able to additionally disrupt the water molecules at the interface. And because of that, these water molecules lack the proper geometry to be able to hydrogen bond with the head groups in these lipids. Okay, so for the sake of time, I'm just going to end here with the conclusion slide. So here are the main conclusions from this talk. The main thing is that if we want to build a synthetic cells, we need to have a way to compartmentalize the cell. So we need to be able to build synthetic organelles. And that is not an easy task, right? Because we need to be able to replicate these complex networks that you see in natural organelles. But the main challenge currently is that we don't have design principles where we can say, if I add this type of lipid at this composition, we're going to get this type of morphology. That doesn't exist. Currently, it's purely phenomenological. And not only that, but being able to predict not just the morphology, but the properties of the interface is still relatively challenging. And in our lab, we rely on IR spectroscopy and molecular dynamic simulations to be able to measure the properties that we would have at different compositions. And so that's what we bring to the table in this community. Let me just end again with the acknowledgement slide. That's my last slide. Let's see if it switches. There we go. I just wanted to mention again, this was done by a student, Kong Zhu, in my lab, was funded by the Army Research Office as part of a MURI, and this is done in collaboration with the Devaraj group at UC San Diego. Thank you for your attention and happy to answer questions.
Thank you very much. There are questions in chat. That was really cool. There are questions in chat. Can you see chat?
Yes, give me 1 second. I need to pull it up here.
If you could read the question out loud.
Okay, yeah, I'll read it.
That's for the recording.
I need to find it. Give me one. I need to find the chat. Oh, here it is. Okay. Avery asked, the sponge face is really cool. Are they more stable than classical vesicles? That is a really good question. Actually, depends how you define stability, right? They are stable in the sense that they don't coalesce or anything. You can actually centrifuge them down and you can work with these droplets. The vesicles tend to, in our experiments, the vesicles tend to aggregate over a period of hours. Whereas this sponge phase, we have not seen any changes in the morphology. So they are probably going to be, yeah, more stable, but we haven't done any characterization, anything in terms of the stability. It's just our experience with the experiments. This actually reminds me of something that I have not mentioned, which is that the limiting factor in a lot of the measurements that we do is the stability of the samples themselves. Each of the spectra that I showed there, it takes about 1 to 2 hours to collect. And we typically have to collect anywhere between 10 and 20 spectra. So sometimes we have to keep the sample in the spectrometer for 24 hours or longer. And in the case of proteins, we've worked a lot with proteins. They tend to aggregate, right? So that's really one of the things that limits our measurements. Sometimes we have to make fresh samples and put them back in the spectrometer and keep averaging for longer. Thanks for that question. The next question is from Amber. And Amber asked, very interesting talk, thank you. Could you use 2D NMR to analyze the properties of other compartmental? Yeah, absolutely, yeah. So 2D NMR is a very powerful technique. The way I see it is not just one technique or another technique, but it's a combination of multiple techniques that is going to be able to give us the insights that we need to be able to make synthetic organelles. In fact, in the Devaraj group, they do X-ray diffraction and neutron diffraction. And obviously 2D NMR would be a complementary technique. The other technique that is useful is EPR spectroscopy. You need to put some spin labels on the lipids, but it's a powerful tool as well. Now, the different techniques, they give you complementary information, right? In the case of NMR, we get timescales on the microseconds and milliseconds, millisecond timescales, whereas in 2D IR, we get picosecond timescales and we get hydrogen bonds, whereas with NMR, you're not able to immediately see those hydrogen bonds. Thanks for that question, Amber. The next question is by Chris. Chris asked, could the sponge compartments be formed in situ as you express enzymes that synthesize the lipids? That would be a question for the Devaraj group. It's a cool idea. I don't know. These lipids are made using your organic chemistry type techniques. They're not necessarily synthetic. But in principle, once the other thing is that the concentrations are relatively high. So currently the way we do it is we preform. So that second part of your question, we preform the vesicles and then add the proteins to the vesicles. But in principle, it could be done in situ. I just don't know very much about how they would be synthetically made, or find a protein or something that would make these lipids. What sample scale do we work with, microliters or milliliters? This is Harvey asking the question. Our minimum sample size is about 20 microliters. We can work with smaller sample sizes. And one of the limitations of this technique is that we have to do everything in D2O. So what that means is that we have to exchange hydrogen, deuterium, exchange the samples, because in water, water absorbs very strongly in this region of the spectrum. So it would not be easy to do the measurements in water. So we exchange everything into D2O, including if we have proteins, we have to exchange the proteins. And sometimes that can be a little bit tricky because proteins can tend to aggregate. So essentially you have to exchange them and relyophilize the proteins so that every single proton in the backbone becomes a deuteron, and then you can make the measurements. Any other questions?
I don't see any questions. I just want to say your talk was amazing for me. I'm a chemist by training, so that's really appealed to me that someone's looking at the molecules.
Thank you. Yeah, thank you again, Kate. I appreciate the opportunity to give this talk.
Thank you, and thanks everyone else. And have a great rest of your day and a great week.
All right. Bye.
Bye, everyone.